So what are the actual steps for getting transcription done? How do we take DNA and make it into an RNA molecule? So, if you remember, here is a DNA molecule. It is going to be double-stranded and it spirals because it's a double helix. So here is some DNA. Now remember, we don't need all of it. We just need some of it. So maybe this section from this section is our gene of interest. For this process, we will use an enzyme called RNA polymerase. So the first thing RNA polymerase is going to do is going to unwind the DNA. So here was our DNA before, and for part of it, it's going to become unwound. So here's the other side. And then here's our gene of interest. It's about from here to here. You have to unwind the DNA because you have to get at one of these two sides. Remember, RNA is single-stranded, so we only have to copy one side. So the next thing that will happen is the RNA is going to attach to an area that is just in front of the gene that is called a promoter. This is the attachment point. This is the other reason why you have to unwind the DNA. That promoter was in here too, but when it's all bound together and spiraled, the RNA polymerase cannot get in there. So now, RNA polymerase binds to promoter. So, my blue marker is dying, so I'm going to get my other one. So here we have our DNA. Here's the other half. Here is my promoter. And here is my gene of interest. So, RNA polymerase is going to start here. The next thing that happens is nucleotides find complements. So remember, there's a whole series of A's, T's, C's, and G's along here. Well, now we're going to have some RNA nucleotides that are going to come in and find their complement. So once they find their complement, then last step, RNA polymerase makes phosphodiester bonds. I'm going to write on the next line. Phosphodiester bonds. So now RNA polymerase will move down this molecule and it will tie each of these nucleotides together. And as we do, we now have created our mRNA molecule. So this here is the transcribed mRNA molecule. This is also called the primary mRNA molecule. That's a 1 degrees mark. 1 degrees is another shortcut way of writing primary. So we have our DNA. RNA polymerase comes in and unwinds the DNA, exposes a gene. It will then attach to the promoter region. Complementary base pairs will fall across from the DNA, but now they're RNA nucleotides, A, U, C, and G. RNA polymerase will then make phosphodiester bonds between them, we now have a primary mRNA molecule. Transcription isn't done, though, until we process that mRNA molecule. So mRNA processing is the second half of transcription. So step one was to create the transcribed mRNA, or a primary mRNA molecule. Step two will be to process the primary mRNA molecule. So if we were to look at a primary mRNA molecule, it's going to be a series of nucleotides that will be a single strand. So this is the primary mRNA molecule. One of the first things that will happen to it is we're going to modify the front. So this side we're going to call the front, this we'll call the end. On the front, we're going to add a guanine cap. So remember, guanine is another one of those nucleotides. So we're going to add a G to the front. So that's a G. This is my guanine cap. So I'll put a G here so you know this. We're also going to add some nucleotides to the end of this molecule. We're going to add a poly-A tail. A stands for adenine. So at the end of this, we're going to add several adenines. Doesn't matter what number you put. I put 4, you can put 3, you can put 6, I don't care. It's many. Poly just means many. And then we're going to cut out certain parts. A primary mRNA molecule is composed of different regions. And these are called introns and exons. So I'm going to put 2 in here. So this region here is an exon. This region here is an intron. This is another exon. This is another intron. Here's another exon. Now you don't have to know where these are, but if I tell you there's an exon, then that's where it's going to be located at. So notice these introns are between exons. When you process the mRNA, you remove the introns. So that means this portion and this portion are going to be taken out of this primary mRNA. Not only do you take those out, but then you just put everything else back in order the same way it was. Then you have an enzyme called a spliceosome. Sorry, that's a C. We'll put the exons back together. So, in the end, we will have the G is still there, it wasn't removed. But then we will have the 3 exons. So we can call this exon 1, exon 2, and exon 3. So here's our 3 exons. Notice that my remaining mRNA is shorter. If you take things out and just squeeze everything back together, it's going to be shorter than what was before. And then we still have our poly-A tail. When we get done, this is what's called a mature mRNA. This mature mRNA is what is going to go to and find a ribosome for the next step, translation.